Have you ever wondered how nearly 2 meters of DNA fits inside the tiny nucleus of every cell in your body? That's the magic of DNA packaging. In this presentation, we'll see how the DNA molecule is organized step by step, from a simple helix to a fully condensed chromosome. Each human cell contains about 2 meters of DNA, but the nucleus is only about 5 to 10 micrometers wide. That's like trying to fit a long rope inside a grain of sand. The cell manages this by carefully folding and coiling DNA so it stays compact, yet still accessible for processes like gene expression. Before we look at DNA itself, here's an overview of an animal cell. Inside every cell, the nucleus acts as the control center. It's where our DNA is stored. Even though the nucleus is extremely tiny, it contains the entire genetic blueprint of the organism. The rest of the cell, like the cytoplasm and organelles, depend on the instructions encoded in this DNA to function properly. Here's where it starts, the DNA double helix. It's a twisted ladder made of 4 bases, A, T, G, and C. The sugar-phosphate backbone gives a structure, and the base pairing keeps it stable. Even before proteins bind, DNA coils a bit on its own, a process called supercoiling, which helps reduce its length. The next level of organization is the nucleosome, the basic unit of chromatin. The nucleosome is the basic unit of DNA packaging in eukaryotic cells. It looks like beads on a string, where each bead is DNA wrapped around a core of 8 histone proteins. The histone octamer forms the protein core of a nucleosome. It's made up of 8 histone proteins, 2 copies each of H2A, H2B, H3, and H4. Around this octamer, about 147 base pairs of DNA are wrapped in 1.65 turns. These histones are positively charged, which helps them bind tightly to the negatively charged DNA, allowing efficient packaging inside the nucleus. These histones act like spools, and DNA winds around them. It looks like beads on a string when viewed under a microscope. This shortens DNA by roughly 7 times. Here you can see that beads on a string appearance. Each bead is a nucleosome, and the string between them is linker DNA. This structure is flexible and active, meaning genes in this region can be easily read and used by the cell. Now, these 10-nanometer fibers coil even further to form a 30-nanometer fiber, often called the solenoid or zigzag model. This level of packaging brings nearly a 40-fold compaction. It's found mostly in heterochromatin, the parts of DNA that are less active. In the solenoid model, the nucleosomes are coiled into a helical structure about 30 nanometers in diameter. Each turn of the solenoid contains around 6 nucleosomes, linked together by histone H1. This higher-order folding further compacts the beads on a string fiber into a denser 30 nanometers chromatin fiber, helping organize DNA efficiently inside the nucleus. This level of compaction is essential for fitting nearly 2 meters of DNA into a tiny cell nucleus. The 30-nanometer fiber doesn't stop there. It forms loops that attach to a protein scaffold inside the nucleus. These loops can be up to 100,000 base pairs long. The scaffold not only supports the structure but also helps control which genes are active by bringing distant DNA regions close together. Finally, during cell division, DNA reaches its tightest form, the metaphase chromosome. 2 identical sister chromatids join at a centromere, forming the familiar X shape. At this stage, DNA is so compact that it's visible under a microscope, ready to be safely divided into daughter cells. DNA packaging begins when the double helix wraps around histone proteins, forming nucleosomes, the basic units of chromatin. Each nucleosome contains about 147 base pairs of DNA coiled around a histone octamer, giving the beads on a string appearance, about 10 nanometers thick. These nucleosomes further coil into a 30-nanometer chromatin fiber, organized in either a solenoid or zigzag pattern. This level of folding is stabilized by histone H1, which helps link adjacent nucleosomes together. The chromatin fiber then forms looped domains, attaching to a protein scaffold inside the nucleus. These loops measure roughly 300 nanometers and play a crucial role in gene regulation and higher-order organization. During cell division, these loops condense even more tightly to form condensed chromatin of about 700 nanometers. Finally, the DNA reaches its most compact form, the metaphase chromosome, approximately 1,400 nanometers wide. This hierarchical folding allows nearly 2 meters of DNA to fit inside a nucleus just a few micrometers across, while still allowing essential processes like replication, transcription, and repair to occur whenever needed. In essence, DNA packaging is nature's way of combining maximum compression with full functionality, a perfect balance between order and accessibility. All this packaging has a purpose. It protects DNA, prevents tangling, and ensures it's compact yet accessible when needed. The way DNA is packed also influences gene activity, playing a key role in development and diseases like cancer. To summarize, DNA packing happens step by step. Double helix 2 nanometer, nucleosome 10 nanometer, 30 nanometers fiber, loop domains 300 nanometer, chromosome 700 nanometer. Together, these levels let our cells store, protect, and use genetic information efficiently. Thank you for listening.